AIDS, and then we discussed the detailed account of another deadly disease, that was cancer. And after that, by now we will be taking another important disease that is pertaining to our accessory digestive gland, that is liver, known as hepatitis. In our today's lecture, we will come to know what hepatitis means and what are the various viral agents, various types of the virus is responsible for development of different types of the hepatitis. But first of all, we should know what actually hepatitis means. You know, so far as the literal meaning of the hepatitis is concerned, hepatic refers to the liver, while as itis refers to the inflammation. So whenever there is inflammation in our liver, but up to that extent, so that what normal metabolic pathways were taking place in the liver, in the hepatocytes, in its biliary, this hepatic system, all such normal pathways get deteriorated, and there is variation in the formation in the pathways of the normal metabolic cycles of our liver. That is hepatitis. So hepatitis simply means inflammation of, it means inflammation of liver marked by variations in normal metabolic pathways, taking place in liver. You know that liver is responsible for detoxification, liver is responsible for protein metabolism, it is responsible for the metabolism of the heme, of the globin, metabolism of various these vitamins, responsible for storage of various vitamins, responsible for glycolysis, glycogenolysis, and responsible for lipid metabolism, and so many other metabolic pathways are taking place in our liver. And it is also responsible for the formation of the bile, which helps us in good fat digestion as well as absorption. When inflammation due to any reason in various types of the liver cells takes place in such a way so that these normal metabolic pathways, which would have been otherwise taking place in the liver, normally they get deteriorated, and that condition is called a hepatitis. So the reason for the hepatitis can be so many things. Sometimes there can be viral infection, sometimes there can be mechanical damage if a person falls down, or he suffers a mechanical injury, he suffers an accident, or if a person is consuming tremendous amount of this alcohol. Such things can also lead to damage to our liver cells or to our liver, and that can also lead to formation of the hepatitis. But normally, when in a normal sense we say hepatitis, then that very hepatitis refers to the disease pertaining to the hepatitis occurring because of infection of various types of the viruses. And the various viruses which are responsible biologically for the development of hepatitis include type A, type B, type C, type D, type E. So the main hepatitis cause is that of the viral cause. The hepatitis which we develop because of viral infections constitute viral hepatitis. So viral hepatitis can be caused due to different types of the viruses. The most common viruses which are responsible for viral hepatitis in the human beings include these viruses: hepatitis virus type A, hepatitis virus type B, hepatitis virus type C, hepatitis virus type D, hepatitis virus type E. First of all, let us have a thorough look over the different types of the viruses responsible for development of different types of the hepatitis in human beings. First of all, let us describe the hepatitis A virus. Hepatitis A virus is actually a virus whose structure is like this. So, this is the hepatitis virus, and in hepatitis virus A, there is no unique envelope. In hepatitis virus A, we can see that there is no unique envelope. No unique envelope. Interior to the so-called covering or the capsid covering of this hepatitis A virus, we can see it is genome, and that very genome is nothing but that is a single stranded RNA. So, it consists of single stranded RNA. Hepatitis A virus consists of single stranded RNA. Remember it. It has no unique envelope. And this very virus has the ability to develop in some persons at least the acute infection. It can lead to the development of acute infection. So far as this very RNA is concerned, which is actually the causative agent of this very acute infection in hepatitis A, this is said to be, this RNA, single stranded RNA is said to be having the ability to use the host cells, ribosomes, for synthesis of its required proteins. Because of such ability of this single stranded RNA, I will mark here it with plus. This plus sign indicates that it has the ability to make use of the hepatocytes, this ribosome, for protein synthesis. So, it is a positive sense RNA, and this very virus is mostly found in tropical and subtropical regions of the globe of the world. It is mostly found in tropical and subtropical regions. That is why travelers before traveling often get themselves vaccinated against this very virus, because it has been found mostly in the travelers who used to visit in the countries of the tropical and subtropical regions. So, in order to protect themselves, they used to get vaccinated on time. And in this type of the hepatitis A infection, we have seen that such persons who are hepatitis A positive, in their serum, they have elevated levels of immunoglobulin G. They have elevated levels of immunoglobulin G. This is a little information about the hepatitis virus type A. Now, there is second type, that is hepatitis virus type B. This is a very deadly virus. One more thing I should write down here. So far as the mode of infection of this very virus is concerned, its mode of infection is oral fecal mode. What do you mean by oral fecal mode of infection? In this type of the infection, such viruses are present in the feces or in the fecal matter of the positive patient. If there is not a proper sanitary conditions maintained by a community, by a state, by a city, by a region, then such positive persons can defecate the fecal matter along with these viruses or along with these hepatitis A viruses. Then with some eatables, especially with the water, these viruses can infect the other persons who will make use of that infected water or that infected eatable, which would have been otherwise contaminated by what? By the fecal matter of the positive person. So, such a type of mode of infection is called oral fecal mode of infection. And this oral fecal mode of infection is the route which is being utilized by the hepatitis virus type A to infect new and other persons. And then we have to discuss another important virus responsible for development of the hepatitis in persons, that is hepatitis type B virus or HPV. So, it is called as, this is hepatitis A virus. And now let us have a look over hepatitis B. This is the hepatitis B virus. When we are having a look over the hepatitis B virus, then we can see that it has a unique envelope. So, external unique envelope formed of lipoproteins, lipoprotein bilayer or lipid bilayered envelope is present in hepatitis B. After that, it also has a capsid covering. Then after the capsid covering, it has its genetic material. And so far as the genetic material of the hepatitis B virus is concerned, it is not the RNA, but it is double-stranded DNA. You know what double-stranded DNA is. Actually, it is not fully double-stranded like eukaryotic cells have double-stranded DNA. It is partially or conditionally double-stranded DNA in comparison to, if we will compare the DNA of the hepatitis B with the DNA of the eukaryotic cells like our cells, then this DNA is not having the exact double-stranded nature. What the type of double-stranded nature of the DNA is present in eukaryotic cells or including human cells and other eukaryotic cells. So, it contains partially double-stranded DNA, and after that it contains capsid coat, and it also contains this unique envelope. And among all the virus, these viroid families which are responsible for development of the hepatitis, this is the only virus or hepatitis B virus which has DNA as its genetic material. This is hepatitis B virus. This was hepatitis A and this is hepatitis B virus. And so far as the mode of infection of this hepatitis B virus is concerned, its mode of infection is, it can infect the persons through sexual intercourse, through blood, and it can also transport itself through the placenta, and it can also infect this developing embryo or developing baby. So, mode of infection can also be prenatal as well. Prenatal as well. So, the persons who get infection of the hepatitis B virus, the mode of transmission for this very virus is through unprotected sexual intercourse with the infected person, and blood transfusion, because it is this type of the virus is also present in the blood of the infected persons. So, blood transfusion, use of the needles by different persons, use of infected needles can also lead to this transmission. Use of such things like blades, like razors, they can also lead to transmission of the virus. And prenatal is also there as a means of mode of transmission for the hepatitis B virus. We should remember it. So, in order to protect oneself from getting infected from the hepatitis B virus, one should take care about all these things of the modus of infection of the hepatitis B virus. And one more thing with the hepatitis B virus is that it can lead to the development of acute as well as chronic or both levels of the infection. it can lead to development of both acute, it can also lead to development of this chronic, it can also lead to development of both modes of the infection. This hepatitis B virus has this very much of the ability. Not only that, it is this very virus which can lead to severe damage in the liver, in some other cells as well, about which we will come to know in a little bit later. And the virus or the hepatitis B virus, actually this virus belongs to a particular family that is known as Hepatinovirus family. It belongs to, you should remember one more thing, that this very virus belongs to this very family, that is Hepatinovirus family. Okay. Now let us have a look over the third type, that is hepatitis virus C type. And this very virus also contains external covering or the envelope So here in hepatitis virus hepatitis virus C, here envelope is also present, here capsid coat is also present, and anterior to the capsid coat there is prevalence of the genetic material. But here the genetic material once again is not DNA, but it is the RNA, single stranded RNA, and this single stranded RNA is once again the positive sense. It is the positive sense because it can utilize the host cells ribosomes for having synthesis of its required proteins, enzymes and other things. So far as modes of infection of the hepatitis C virus are concerned, they are the same as are the modes of infection for hepatitis B. So mode of infection are same as hepatitis B virus, isn't it? And this very virus has also the ability to develop acute or chronic symptoms. It has the ability to develop acute or chronic conditions. Then there is another virus, that is hepatitis virus type D. This hepatitis virus type D, it is actually belonging to another family of the viruses. That very family is called as virusoid. It belongs to virusoid family of viruses. And this hepatitis D virus also has its unique envelope, lipid bilayer envelope here, and capsid coat, then RNA. It also has envelope, it also has capsid coat, and then it has single stranded RNA. Remember one important thing here, that the single stranded RNA which is present in the hepatitis D virus, that is of negative sense. This is not of positive sense, this is of negative sense. What does that mean? That means this very virus is not having the ability to make use of host cells, ribosomes, for having its required or needful protein synthesis. So this very single stranded RNA of the hepatitis type D. This is HPD. It is of negative sense, remember it. And for becoming infectious, for achieving the ability to complete its life cycle, to develop the hepatitis, it needs some help. Which help? It needs the presence of some other antigen which is being synthesized by the hepatitis B virus. That very antigen is hepatitis B surface antigen. It requires hepatitis B antigen to make use of its RNA in the host cell's cytoplasm and to make use of host cells these ribosomes for having normal protein synthesis and to develop hepatitis in the patient. So, it needs a co-infection. It needs a co-infection. It means the persons who are infected with hepatitis B, they have the chances to develop the hepatitis D as well. And here the modes of transmission are again the same. What would have been the modes of infection for type B or type C? So, modes of infection are again sexual means, the blood, etc. In this hepatitis type D, we should remember one more thing, that here the persons who are positive with the hepatitis D, they show the elevated levels of immunoglobulin G and immunoglobulin N. They show elevated levels of, here I will write down immunoglobulin G and immunoglobulin N. And for this virus, there is requirement of this very antigen which is being synthesized by hepatitis B virus. So, co-infection of this very virus for the hepatitis B is mandatory to develop hepatitis among some persons. However, in some immunosuppressant persons, it may lead to directly development of the hepatitis. And after that, there is next hepatitis, that is hepatitis type E virus. Previously, it was not considered to be the type E or type E of the hepatitis was not considered for being an infectious or viral infectious hepatitis disease. But later on, people came to know that there is another virus known as hepatitis type E virus, which is also responsible for development of the hepatitis in some persons as well. And so far as this very type E hepatitis virus is concerned, it has the ability to develop acute infection. It has mostly been seen for showing it is present in immunosuppressed persons and pregnant women, where it can lead to the liver damage. And so far as its structure is concerned, it is not having any envelope. No envelope is present in type E. It has no envelope. And it has a single stranded RNA, which is again of positive sense, which means it can utilize the host cell machinery for synthesis of proteins. And in this type, there is always rise in the immunoglobulin type M. there is raised level of IgM, and this has been mostly found in Asia and Middle East regions, mostly found in Asia and Middle East regions of the globe. But it has been seen that even some domestic animals or some other types of the animals can also be the modes of can also be acting as the vectors for the modes of transmission of this hepatitis type E virus. So this was all about the different types of the viruses which can lead to hepatitis. Though hepatitis can be in a person because of some mechanical injury as well, because of overuse of drugs, because of overuse of the alcohol, and some other conditions can also lead to hepatitis. But so far as viral hepatitis is concerned, it is because of these different strains of the viruses which have the ability to develop hepatitis among the persons at its acute level, at its chronic level, at its elevated level, which can lead to the deterioration of the liver and which can lead to transformation of some hepatocytes into the cancerous types of the cells. Now, let us have a look over the mechanism which is being operated over the cells of the liver with which hepatitis is developed, and let us have a look that in case of some acute or the severe hepatitis, what happens within the blood, within the liver, within some other organs of the patient. If this is the liver, say this is the liver, and a person has been infected by, say here it is type 1, here it is 2, it is 3, it is 4. Now, actually I have used this 1, 2, 3 and 4 instead of type A, type C, type T, type E. So, this 1, type 1, type 2, type 3, type 4. When this type of virus comes, after using their respective routes, if it has infected a person, this virus ultimately reaches up to our liver. And let us consider that this is not the whole liver, but this is as a single particular liver cell, that is the hepatocyte. Let me write down here as if it is the hepatocyte. Now let us see what will happen to the hepatocyte after it has been infected by these strains of the viruses responsible for development of the hepatitis. When they will infect it, well, they will infect the hepatocyte, they will shed their envelope if that is present, they will shed their capsid coat if that is present, and they will lead to entry of their respective single stranded RNA into what? Into the cytoplasm of the hepatocyte. And in the cytoplasm of the hepatocyte, we have seen that most of such viruses are of positive sense, so they are having the ability to make use of host cells ribosomes. Say this is the host cell ribosome. So once this viral RNA has entered into what? Into the hepatocyte, it has used the host cell's ribosome, and then it has started the synthesis of various proteins, isn't it? And the various proteins which have been synthesized by this very RNA through the process of translation will constitute the capsids, the polymerases, isn't it? The antigens. They will include, I will write down here, capsids. They will also synthesize polymerases, isn't it? They will synthesize antigens, different types of antigens, different types of capsids, different types of polymerases, isn't it? And some many other types of the proteins will be synthesized by the RNA once it has entered, once it has got its entry into the hepatocyte. After these different types of the proteins have been synthesized, these proteins may be transported up to the Golgi apparatus. In the cytoplasm, we have another thing that is Golgi apparatus. GA I will write down here, and in the Golgi apparatus there will be packaging of these very proteins, and the packaging will take place. This is the packaging of proteins in the vesicles. So now here these are the vesicles in which this very RNA, this very viral RNA has put its synthesized proteins, and from these vesicles the respective substances, antigenic substances, polymerases and other things can get their entry into other cells, can get their entry into the biliary system, can get their entry into the blood vessel or into the blood. So after the synthesis of various types of the proteins, whether that is antigens, that is polymerases, that is capsid like proteins, isn't it? After their synthesis, they will be transported and they will be utilized for putting more and more damage to the liver or to the new hepatocytes. They can reinfect. Here I will make some arrows which will show reinfection, isn't it, of the hepatocytes, and they can also lead to lysis of hepatocytes even. They can also lead to lysis of hepatocytes. When the lysis of hepatocytes, then liver cirrhosis can also be there, but it is the first stage in the different types of viral infections to our liver. Sometimes when a person gets infected with this very deadly hepatitis B virus, say here, now this is hepatitis B, isn't it? When it attacks the hepatocyte, it directly gets entry when the DNA, since you know that the DNA is the genetic material in the hepatitis B virus, and when it gets entry into the hepatocyte, then this very double-stranded DNA, see here it is the double-stranded DNA, it penetrates itself to the nuclear pore complex, and there it uses the host cell's transcriptional machinery for having replication of itself. So this very DNA, which was the genetic material of the hepatitis B virus, if it infects a hepatocyte, it incorporates itself into the nucleus of the hepatocyte, then it resynthesizes the new DNA of itself, and when new DNA is synthesized, that DNA can lead to formation of RNA once again, and that RNA can use this route which was used by the RNA of these types of the viruses. But that RNA has another option, since this hepatitis B virus, it is a virus which belongs to which family? Hepatinovirus family. It has another important enzyme known as a reverse transcriptase. By making use of reverse transcriptase, it can again synthesize the DNA, and then that DNA can utilize this very route, isn't it? And after the lysis, after the degradation of that very hepatocyte, that DNA can re-enter into the new hepatocytes, new hepatocytes and new hepatocytes. So, this hepatitis B has the ability of using this route and this route as well. We should remember it, isn't it? This all happens in the first stage of such viral infections, isn't it? Then, once the immune system of the concerned patient comes to know that my hepatitis have been attacked by these different strains of the viruses, it begins to respond. How the response will take place? Let us have a look. You know, our blood contains different types of immunogenic cells. One of the important immunogenic cell is known as cytotoxic T cell. One of the important cell is known as in the blood. There we have an important cell that is known as cytotoxic T cell. When this very cell comes to know that my hepatocytes have been attacked, it first responds. How it responds? It synthesizes an important proteinaceous substance known as CD8. CD refers to cluster of differentiation, CD8. By making use of CD8, it can recognize the MHC of, it can recognize the MHC receptor of the concerned hepatocyte. Here it is MHC. MHC refers to major histocompatibility complex. If you have went through my all lectures of the immunology, then it would be very easy for you to understand what this MHC stands for and what are the conditions in which it is being synthesized. And I have already told you that there are different types of MHC receptors or MHC proteins. The MHC which is being synthesized or which is present over the surface of the damaged hepatocyte, which has been damaged because of this very viral infection, this very type is called as MHC1. So, actually it is the MHC1 which is present over the surface of the damaged hepatocyte, isn't it? And it is this CD8 protein which has been synthesized by the cytotoxic T cells, which will recognize this very damaged or infected hepatocyte because of presence of MHC1. So, this can recognize this. When a recognition takes place, after complete recognition takes place, that when this comes to know that this very cell has been infected, then it begins to induce an important process known as apoptosis. So, when it has recognized it, then it will start apoptosis. And you know apoptosis is what? It is programmed cell death. So, it will quickly guide that infected hepatocyte to die, to get killed as soon as possible, so that no new hepatocytes will be infected from the viruses which have already infected this very cell. This is the first thing. Second thing is that when apoptosis is on its way, then the respective or the damaged region of the hepatocytes can induce the synthesis of other substances from this very cell. It can synthesize another proteins known as perforins. When it has recognized a particular hepatocyte or hepatocytes that they have been infected by different these hepatitis viruses, then it begins to synthesize another format of the proteins known as perforin type of the proteins. And it is because of this perforin type of the proteins which have been synthesized by the cytotoxic T cells that they will lead to perforations of the hepatocytes. When perforations of the hepatocytes takes place, then activation of another type of the protein, these cells takes place, that is macrophages. They lead to perforations. When perforations of the hepatocytes takes place, then their perforations will lead to release of some other factors like release of, I will make that here, interferons. It will lead to release of interferons. When different types of interferons are released, these interferons come and they are transported, they come and they are transported via blood. It is a blood vessel. So now the new interferons which have been synthesized or produced by the damaged part or the damaged tissue, these interferons will come in the form of alpha type, in the form of interleukin 1 or 2. Alpha, all of them will come and they will induce brain, isn't it, to release some particular prostaglandins like PGE, which will lead to induce the vomiting center of the brain, and which will lead to a regurgitation of the food, and that regurgitation of the food will lead to vomiting, which will lead to increased cramping movements or peristaltic movements of the gastrointestinal tract, especially this very gastric region, and which will ultimately lead to diarrhea. It can lead to diarrhea. And sometimes when there are increased interferonic secretions from the respective these damaged tissue, sorry, these are actually interleukins, these are interleukins. When there is increased release of interleukins, then that increased release of the interleukins will lead to a release of the prostaglandin type E, and this very release of the prostaglandin type E will induce this gastric center of the brain, of the brain stem, to guide the gastrointestinal tract for vomiting, for diarrhea, and this can lead to the loss of water, this can lead to dehydration, this can lead to the imbalance in the electrolyte system which would have been present in our blood and in our cells. So electrolyte imbalance will take place because of this electrolyte imbalance, isn't it? Dehydration dehydration, thickening of blood. When more dehydration will take place, that can lead to thickening of blood as well. Besides that, this very prostaglandin E release will also lead to influence the emetic center of the brain. It will also influence the emetic center of the brain to exhibit these changes, and it will also induce the hypothalamic region of the brain, hypothalamus, to go for more and more production of the heat, leading to fever. See what happens when this very cell comes to know that this very damage has taken place. It will release CD8, it will release porphyrinus, and that will indirectly induce these cells for having synthesis of the interleukins, which will indirectly change the brain cells for having changed synthesis of the PGE like prostaglandins, and indirectly there will be change in the emetic center, and there will be increased vomiting, there will be increased diarrhea, which will lead to dehydration, electrolyte imbalance, and which will lead to the thickening of the blood and so many other problems. And you can clinically correlate these conditions, that what will happen to the patient at that time, and it will also lead to increased body temperature, because it will also influence the heat center of our brain, and as a result of which the person will be suffering, or he will develop the high intensity fever as well. And besides that, when the fever like conditions, vomiting like conditions, and other conditions are taking place in the person, if the infection continues, if the person will be kept untreated and the infection prevails there and the damage prevails there, then the damage will go little ahead. And what other collateral damages will take place in the respective person? Let us have a look. Since you know that it was the liver which was responsible for various metabolic pathways, and for that this liver has many types of the enzymes to maintain a balance over the metabolic pathways, like AST is there, AGT is there, CGT is there, isn't it? But now there will be, when damage of the hepatocytes will take place, there will be elevated levels of some unique enzymes, which the elevated levels which can be seen in the serological tests of the patient. So when damage of the hepatocytes will take place, that damage can lead to increased level of this enzyme AST, ALT, alkaline phosphatase as well. Their level increases in other damages. Even CGT level, this is increased, this is increased, this is increased, this is increased. So their increased level, after we conduct some serological tests to the patients, they will also indicate that about what level the damage of the liver has taken place because of the infection of these respective viral agents. Not only that, in the patient, when the patient is untreated still and the infection goes ahead and the new viruses are coming out, they are damaging the new hepatocytes, they are again damaging the new hepatocytes, they are damaging the new hepatocytes, then in that person, because of changed metabolic pathways, the biliary system is also damaged. The biliary system is also damaged. We know that in the so far as the biliary system of the liver is concerned, there was taking place synthesis of bile, and bile was containing two important things. Number one, salts, isn't it? And number second, pigments, especially bilirubin. And this bilirubin was then being converted by making use of some bacterial enzymes in the gastrointestinal tract. That was being converted into the citrocobalamin and other formats of the pigments. But when the damage of the hepatocytes takes place, then the salts or the salt level of the bile, that gets changed. Some salts increase, some salts decrease, isn't it? When their levels are increased or decreased, then the part of the digestion which otherwise would have been normal because of the bile salts, then that is damaged. So another thing will take place, that is damage to digestion process in duodenum. Bile salts were mostly associated for regulating the pH, for the formation of the micelles, for fat digestion, for lipid digestion, but all such things will not take place now normally. And so far as the bilirubin is concerned, you know that the bilirubin was being synthesized from the heme part by the hemoglobin by different enzymes, from the heme part of the hemoglobin from different enzymes of the liver. But now, when the damage is more and more to the hepatocytes, then the bilirubin metabolism is not taking place normally, and then there can be increased levels of bilirubin unwantedly, citrocobillinogen, urobilinogen, and that can lead to formation of dark or clay colored feces. You know that citrocobillin was responsible for imparting color to the feces, but when citrocobillin synthesis is increased unconditionally, unwantedly, then that can lead to development of dark or clay color to the feces. And in that person, this urobilinogen will be now synthesized more and more, and as a result of which that person's urine will also become dark. Isn't it? Not only that, the bilirubin level, conjugated as well as conjugated form, the bilirubin level, both conjugated as well as unconjugated, that will be increased. When bilirubin level in the blood will be increased, then this very bilirubin of both hepatitis forms, now that very bilirubin has the ability to get accumulated in our eyes, that can lead to formation of pale eyes. Not only that, that can also get accumulated on our hands, palms, soles, peripheries. So this jaundice-like condition will be developed there. Pale hands as well. Isn't it? First, let me check whether my video is being recorded or not. Yes. So you have seen that when the infection proceeds there, then the damage goes ahead. Then the damage leads to increased level of the bilirubin, increased level of these very enzymes, and the discoloration of the fecal matter, discoloration of the urine, and discoloration of the white of our eyes, discoloration of the palms, of the soles, of the skin surfaces, of other tissues, isn't it? Not only that, their increased level can also lead to sometimes the blockage of the cystic duct or the bile ducts, which will otherwise also lead to putting up the more and more pressure over the liver. And then these increased enzymatic conditions and the increased pressure of the bilirubin of the bile or the hepatocytes or the biliary system of the liver will lead to inflamed liver, and now the size of the liver will be increased, and the condition will be called as hepatomegaly. The condition will be called as hepatomegaly. So hepatomegaly will also be the outcome of some chronic infections of some hepatitis viruses. Not only that, when the blood will become more and more thick, then the blood and the fluid sometimes may get retained in the joints of the bones, and then that can lead to development of arthritis as well. So the patients may also develop arthritis, isn't it? and sometimes the patients can also develop the vasculitis with respect to his blood vessels. Not only arthritis, sometimes the patients will also develop the vasculitis. The patient can also develop vasculitis. After the patient develops vasculitis, inflammation of the blood vessels, then in that condition, so far as the regions outside the heart or in the different chambers of the heart are concerned, we have pericardium, we have endocardium, we have myocardium, and we have coronary blood vessels. In case of the vasculitis, the coronary blood vessels, they may also get damaged. The blood vessels which will supply blood to the heart and the blood vessels which are directly associated with the heart, they may also get damaged, and the fluid which is called as the pericardial fluid, its thickening or presence of different toxic substances synthesized by the viruses or the viral antigens, they will also lead to the over synthesis of such fluid, leading to another condition, inflammation of the heart muscles, leading to increased size of the heart or cardiomegaly or cardiomegaly. Not only this, when these changed conditions and when these antigens are present in the blood of the infected persons, they will reach up to the kidneys as well, and they will deteriorate the normal this filtration process of the kidneys, and they can lead to nephritis as well. Nephritis which can impair the normal filtration process. By our kidneys. So, you are seeing in some chronic infections what damages can be experienced by the patient's body with respect to his this liver, with respect to heart, with respect to brain, with respect to duodenum, with respect to stomach, with respect to blood, isn't it? With respect to coloration and with respect to this nephric filtration and many other things. And besides that, sometimes in more acute conditions, in more chronic conditions which are known as the convalescent stages, there is where there is increased levels of the antigens, these immunoglobulins and other proteins and interleukins and other substances. There is damage to the synthesis process of the platelets as well as the neutrophils, leading to decrease in concentration of neutrophils. Neutrophil concentration will be decreased, and platelet count is also decreased in the patient's blood. So, more chronic convalescent stage of some of the hepatitis will lead to decreased neutrophils. It will also lead to decreased platelet count. Then you can yourself assess the clinical these symptomatic conditions which will be otherwise experienced by the concerned patient. And when there will be a decreased prevalence of the neutrophils or the platelets, you can yourself assess that the patient will not be in a position to clot his these injuries or injured vessels, and patient will not be in a position to cope up with some infections which would have been otherwise managed by the neutrophils or neutralized by the neutrophils. Meanwhile, in these convalescent stages, we can see that there is increased level of the lymphocytes. Lymphocytes have to neutralize these conditions. For the neutralization process, there should be increased level of the lymphocytes. So, in that patient serum, we will see that there will be increased level of the lymphocytes. And besides that, when the synthesis of these substances takes place, when we have seen that the porphyrin is very synthesized, when the more damage has taken place to the hepatocytes, then the release of the porphyrins, then the release of some factors from the hepatocytes can also lead to induce another cells which are known as macrophages. They will also lead to induction of other cells known as macrophages to have their respective action, isn't it? When there is increased synthesis of the macrophages, then macrophage increased synthesis will lead to damage in the biliary system, will lead to damage in the damage of the other tissues of other regions of the liver. They will also lead to liver cirrhosis as well, and they will also lead to sometimes even the deadly condition, that is fibrosis condition and development of the cancers. So their overaction will lead to two more conditions, that is fibrosis, chronic liver damage, convalescent stage, or it will also lead to, since you know, when the damage of the hepatocytes will take place, then there will be increased synthesis of the lymphocytes, increased synthesis of the macrophages, increased action of the cytotoxic T cells, and sometimes the failure of the hepatocytes will take place with respect to their regulation of the cell cycle. Then their increased cell cycle, increased reproductive phenomena will take place, and that will lead to conversion of normal liver cells into the cancer cells. So sometimes the hepatitis can lead to development of liver cancers. So we have seen in acute and chronic conditions that that different sorts of the damages will be there with respect to the patient after he gets infected with different types of the viral strains. And we have seen in the different phases that the level of the damage varies. We have seen in the first stage, which is known as the prodromal stage. We can divide the whole damage stages into different stages. First very stage is known as, I don't have space now, first damage stage is known as prodromal stage. In the prodromal stage, this infection takes place, this pathway is there, this pathway is there, and not more damage is not to the liver, and there is no more damage to the kidneys, no more damage to the heart, no more damage to the duodenum and the brain and other things. But in the second part, that is known as icteric phase. Icteric phase of the damage, we have seen that there is change in the level of the enzymes, there is change in the their bile synthesis, there is synthesis of the interleukins, then there is misguidance of the brain and brain stem of the hypothalamus, there is fever-like conditions, isn't it? There is vomiting, there is nausea, there is diarrhea, there is electrolyte imbalance, there is thickening of the blood, etc. And after that, in which more complex conditions we have seen with respect to the conversion of the normal cells into cancer-like cells, with respect to the fibrosis. That very third stage is called as convalescent stage. Convalescent stage. With this, we have concluded our today's lecture, in which we came to know about the different viruses which are responsible for development of the hepatitis, in which we came to know about the hepatitis type A virus, type B virus, type C virus, type D virus and type E virus. And we also came to know little bit about the structure of these respective viruses and the presence of the genome and the type of the genomes in the form of the RNA or the DNA in the respective viruses. And then we came to know about the different acute and chronic conditions these experienced by the patients, in which we came to know about the prodromal stage, about the icteric stages, about the convalescent stages, in which we have seen that at which level the liver hepatocytes are damaged. Then other cells are damaged, then variation in the biliary system takes place, variation in the secretion synthesis of the bile salts, bile pigments will take place, and damage to the liver, damage to the biliary system, damage to the this protein lipid digestion, damage to the blood, content of the electrolytes, content of the water level, all such changes we have seen. And we have also seen the changes in the most convalescent stages which were associated with some other important organs like brain was there, kidney is there, heart is there. So we have seen when most these chronic conditions of some types of the hepatitis B and the hepatitis C viruses takes place to a patient, then we have seen that the damage can go beyond that of the liver. The damage can be up to the blood, it can be up to the brain, it can be up to the kidneys, it can be up to the duodenum, it can be up to the stomach, it can be up to our palms, of the soles and the eyes and other things. So with this we have concluded our today's lecture, and in our next lecture of the biology and human welfare chapter we will be discussing some of the severe conditions developed in some patients by hepatitis B virus. So till we will take our next lecture. If you have got something good in my today's lecture, I request you to get my channel subscribed so that I will get boosted with your good comments, and I will change the expression of my lectures, the expression of my this giving process in my teaching. Till now, stay blessed and God bless you.